Good afternoon. We're going to talk today about how to measure enzyme activities in natural waters. And so there's two components to measuring enzyme activities. The first thing is we need to take our natural water sample and add what's called a fluorogenic enzyme substrate. So this is a chemical that creates fluorescence when an enzyme is present. We're going to add that to our natural water sample, and we're going to measure the rate of fluorescence production over time. That is, how fast fluorescence is produced over time in a sample. And that's the first thing. That tells us the activity of the enzyme. And the second thing that we're going to do is calibrate. We're going to do what's called calibrating the sample, which is we're going to add a known amount of fluorophore. So that's a chemical that doesn't interact with enzymes. It's just a fluorescent chemical. And we're going to add a known amount of it and see how much fluorescence that produces. So that's going to allow us to translate between the fluorescence units that we're going to measure in the enzyme activity measurement and the actual number of molecules that are being produced by the enzymes. And there's a third layer of complication, which I'm going to get into in a minute, which is we want to investigate the kinetics of the enzyme. So kinetics is the study of how the rate of a reaction is related to the concentration of the reactants. And so the kinetics of enzyme activity tells us a lot about the nature of the enzyme that we're looking at. So the way we're going to do that is we're going to measure activity of enzymes in different samples at different levels of substrate concentration and see how the enzyme activity, the rate of fluorescence production, is related to the concentration of substrates that's present in the sample. So that's sort of three different things. The first thing we're going to do is just look at the enzyme activities. But before we do that, I want to get our standard, our fluorophore standard that we're going to use for calibration out of the freezer so it can start to thaw. So I think you guys have your substrates in a freezer. You're going to want to thaw them before you use them, obviously. There's a trick to doing this, actually, because it's going to look like these are dissolved in water, which has been frozen into ice. But that's actually not the case. They're not dissolved in water. They're dissolved in a chemical called DMSO, or dimethyl sulfoxide. There's two kind of funny things about dimethyl sulfoxide. The first one is, although it's not a dangerous chemical by itself, you want to be a little bit careful because it has the property that it makes your skin more permeable to any kind of compound, including anything that might be toxic. And so when you're using DMSO, you always want to wear gloves because you don't want to get DMSO on your skin. If you do, like I said, it's not terribly dangerous by itself. You don't have to freak out. You just want to wash your skin if you get any on you. You don't have to worry about it. But you do always want to use gloves. So first, I'm going to put on my latex gloves. Small fraction of people in the world have an allergy to latex. If you have an allergy to latex, then don't use latex gloves. So now I'm going to get what's the fluorophore standard. The fluorophore is called aminomethylcoumarin, or AMC for short. I'm going to get it out of the freezer. Keep it in a little vial like this, which is called a centrifuge tube or a falcon tube. The funny thing about DMSO is that, like I said, it looks like ice, but it actually has a freezing point instead of 32 degrees Fahrenheit. It's got a freezing point of about 55 Fahrenheit or so. So that means you actually have to get it fairly warm before it will melt. So the way that I'm doing this is that I am going to put it in this little beaker of warm water that I've set out, and hopefully by the time we're ready to use it, it will be thawed out. So now we're going to move on to the main task, which is measuring enzyme activities. And specifically, we're going to measure enzyme activities at different concentrations of substrates, and because we want to control for all of the variables in our experiment, we're going to measure enzyme activities in a live sample. So this is some water that I took out of the Tennessee River, which is close to here, and in a what's called a killed control. So I've taken some of that same sample, I've put it in this very scientific water boiler that I bought from Walmart, and I boiled it for 5 minutes. And that destroys all of the enzymes that are present in the sample. So I'm going to measure the enzyme activity in that sample. Hopefully it'll be 0. If it's not 0, we know that we're getting something that looks like enzyme activity, but is due to something other than actual enzymes. So what we need to measure enzyme activity is we need our water sample. We're going to use 1 milliliter of that. We need our enzyme substrate. I'm going to use different volumes of that, but small volumes, because we don't want to change the total volume of the solution too much. And finally, we want to make sure that the pH of the sample stays constant during the experiment. And so we're going to use something called a pH buffer. And this is just a liquid that resists changes in pH. In this case, I'm using a buffer using phosphate salts, so this is called a phosphate buffer. I've pH'd it to a value of 7.6. So during the experiment, the pH in our cuvette should stay constant at 7.6. So we're going to do the experiment in these little vials that are called cuvettes. The point of these vials is that they are transparent to ultraviolet light, which is being used to excite the fluorescence, and they're also transparent to all the visible ranges of light. So what's going to happen is the fluorimeter is going to shine a light in the front of this cuvette, and then the fluorophores are going to emit fluorescence in all directions, and then the fluorescence detector has a little electric eye, a little photodiode on the side that's collecting the light that goes out the side. So we're going to add first 1 milliliter of our sample to each of 20 of these cuvettes. So like I said, we want to do this measurement at different concentrations of substrates. And you can see that this is the rack that the cuvettes come in from the factory. And you can see that I've labeled this. On the rows, I've labeled 1L and 1K. It's a little hard to read because it's a small piece of styrofoam. But that's for substrate number 1 live and substrate number 1 killed. When I do a real experiment, I measure multiple different enzyme substrates to look at different enzymes. So we'll have substrate 2 live, substrate 2 killed, substrate 3 live, substrate 3 killed, etc. Then across the top, I've written the different volumes of substrates I add, and we can do a conversion later on to calculate the concentration of the enzyme substrate given the volume I added. But this is volumes in microliters. It's 0, 5, 10, 15, 20, 25, 30, and then I've done 3 times 40 because I want to measure the, I want to have replicate measurements for the highest concentration. So I have 3 cuvettes where I'm measuring a volume of 40 microliters of the substrate. So the first thing I'm going to do is I'm going to add my sample. I collected my sample in just a little plastic box called a cuvetainer like this, and then just for convenience, I poured it out into this 50 milliliter centrifuge tube or Falcon tube. And like I said, I also boiled a subset of this water and also poured it out into this centrifuge tube. And I'm just using these centrifuge tubes because they're more convenient to get. So I'm going to take my 100 to 1,000 microliter micropipetter. That's 0.1 to 1 milliliter. I'm going to make sure it's set to it says 1,000 on here. That's 1,000 microliters or 1 milliliter. So that's how much sample I want to add. And I'm going to take my pipette box. You don't want to get any dirt in the pipette, in the box of pipette tips, so we always just open it up, take out a pipette tip, and close it up. And notice that when I took out the pipette tip, I didn't jam it on real hard. There's no need to do that. I just sort of put it on gently, just enough force that it'll stay on. So first I'm going to take my live water and add 1 milliliter very carefully to each cuvette. I'm actually sit down to do that. I find in a lot of cases, you don't want to add something to the same cuvette twice. So a lot of times I'll put my little finger on the column that I'm adding to. And notice that I'm just drawing the water up fairly quickly, but also gently. I'm not letting it spring up, and I'm making sure there aren't any bubbles coming up into the pipette tip. And then the same thing, I expel it, not fast so that it splashes, but relatively quickly and steadily. So each of these cuvettes gets 1 milliliter of live sample. And then I don't want to contaminate my killed control with any live water from the pipette tip, so I'm going to throw away the pipette tip, and we can just do this by pressing down hard onto the button. Oh, sorry, this one, where it ejects. And I've let this kild control cool down because we don't want to put it in when it's hot. That might cause the substrate to break down and emit more fluorescence than it's supposed to. A lot of times, I always count either in my head or even out loud to make sure that I am on the correct cuvette, because you really don't want to make any mistakes pipetting. It's very easy to do, and it will screw up your experiment, and you won't find out until you look at the data. So you want to be very careful with how you're pipetting. Make sure you pipette the right thing into the right place. It gets pretty complicated with this experiment. And you notice that I'm always trying to keep the pipetter as vertical as possible, and I'm absolutely making sure not to let the pipetter go upside down, because if any water gets in the pipetter, that can ruin it, and that's really expensive. So now I'm going to do the same thing with the buffer. I'm going to add 100 microliters of buffer. I'm always paying attention when I'm adjusting the pipetter to make sure that I don't adjust it outside of the bounds of the limits of the pipetter's volume range. The 100 microliter pipetter, or the 20 to 200 microliter pipetter, takes a smaller tip. Use the small tip, press it down. I've got my vial of phosphate buffer, and you notice that I always label all of my vials so that I don't get confused about what's in what centrifuge tube. And again, I'm going to add 100 microliters to each of these cuvettes. Alright, so we've got all of the samples and the phosphate buffer added. So all we need to add now is the substrate, the enzyme substrate. And right now I'm going to be using an enzyme substrate for an enzyme called leucine aminopeptidase. The substrate is leucine 7-amido-4-methylcoumarin, or LU-AMC. The point is that it looks for enzymes that break an amino acid called leucine off the end of a protein. But the key thing here, like I mentioned before, is that we're going to add different amounts, different volumes of substrate to almost each vessel. And the reason that we're doing that is because we want to see how fast the reaction goes at each different concentration of substrate. And that tells us about the enzyme kinetics. So I'm going to use this 5 to 50 microliter micropipette. In general, when we do something like this, when we're adding different volumes, we want to use the same micropipette because each micropipette is a little bit different. And so if it's a little bit inaccurate, this micropipette, at least it will be inaccurate in the same way in each sample. So I'm going to adjust it to 5 microliters. And notice that the lowest concentration of substrate that we add is 0. So the first cuvette is going to have 0 substrate in it, which is because we want to know whether fluorescence changes in the absence of any fluorogenic substrate. So we just skip that first column and draw just a little bit. 5 microliters is just a very small amount, and I'm not sure if you'll be able to see on here, but you want to get that 5 microliters out without touching the sides of the cuvette, because there's obviously a live sample in there, and you don't want to get any of your live sample in your enzyme stock, or your substrate stock, or else all of the enzyme substrate will get hydrolyzed and it won't work anymore. So when I'm doing this and not talking at the same time, I'm always very careful to pay attention to the volume that I'm adding and the row and column that I'm adding it to, because this is the easiest place to make a mistake. That's also the reason that I like to write the volumes that I add onto the little styrofoam box, because it helps me remind me where I am. And of course, as I'm adding this, as I'm putting the micropipetter in here, I try not to touch the walls of the micropipetter to the walls of the centrifuge vial, but I can't totally do that, so I'm going to wipe this down afterwards. So I'm adding up to 40 microliters. The stock that you all have is 10 millimoles of enzyme substrate per liter, and so if we're adding 40 microliters of that, that comes out to 400 micromoles of enzyme substrate per liter at the highest concentration. And for these kinetics measurements, we want to get a few concentrations on either side of the what's called the Km, so the sort of value of which it starts to be saturating. And usually for peptidases, that's around maybe 100 or 200 micromoles per liter. So that's why I've chosen this concentration. Stop that. So now that we've got everything we need in each vial, the last thing to do is put a cap on each vial. You want to make sure to do this with clean hands. If you think you've got substrates or something like that on your hand, you'll want to change your gloves, because you have to put your hand into this big bag. And you don't want to get substrates or anything else all over these caps. So now that we've got the tops on all of the cuvettes that we're going to be using, we want to mix them up. All the chemicals that we've added are nice and well mixed, and the easy way to do that is we just put the top on, hold it on, and turn it upside down a couple of times. So now that they're nice and well mixed up, we are ready to measure the fluorescence. The way we do that is with this fluorimeter, which is just about exactly the same as you all have. Real easy to use. All we have to do is turn it on. It makes us wait 5 seconds. Before we measure any samples, we want to take the solid standard out of this little case here. So the solid standard is just a reference to tell us whether the instrument response, or the amount of fluorescence it reports, is drifting or not. By the way, before we measure this, we want to make sure that the only thing we have to do is make sure that it says UV down here and not blue. If it says blue, press AB and make sure it says UV. And then after you do that, there's a bunch of buttons on here, but the only ones you want to pay attention to are on, off, AB, and read. So we've got the solid standard in there. We're ready to read the fluorescence. It tells us for about 5 seconds that it's reading, and it comes out to 812.0. So we're going to write this down in our lab notebook. Of course, whenever we do any kind of scientific experiment, we always make sure to record everything we're doing in a lab notebook. Every page of the lab notebook should have a date on it right there. It should have a title describing what that page is about. And then in this case, I've described sort of the general purpose of what I'm doing and the procedure of how I did it. And then I've got this table set up to record the actual data. So the way we want to record data is we take out the cuvettes one at a time. We try to keep them in the dark the rest of the time. We line up the cuvette like this so that the flat side is facing towards us and the indented sides are on the sides. Put it down firmly into the fluorimeter, but again, don't force anything. One really important thing is we don't want to get any liquid into the cuvette cavity where the fluorescence is red, because that'll cause all kinds of problems. Close up the top and press read. Now when we do that, I've got a clock right here. It's got the time. I always use 24-hour time. So I'm going to record the time in hours and minutes along with, get that? Along with the fluorescence value. So the time is 1531 and the fluorescence is 194.8, and the sample was the live 0 microliters sample. So then I'll just go down the row, try and measure these nice and quickly. Time is still 1531. Fluorescence on this one, this was the live 5 microliters, and the fluorescence was 266.5. And I'm going to measure the solid standard once for every row that we measure. So I don't measure it every single time that every row of cuvettes that we measure, I measure the solid standard. For the most part, it doesn't drift too much, but we always want to know if it does start to drift. And it's not surprising that the fluorescence is going up even at the very beginning of the experiment before the enzymes have had time to do much, because there's a little bit of background fluorescence to the enzymes, the substrate that we added. And that means that the background fluorescence is going to go up a little bit. But remember, we're not looking at the value of the fluorescence, we're looking at how fast the fluorescence changes over time. So I'm going to go through this whole row and then the whole row of kilograms and record the time and the fluorescence for each one, and that will be my set of starting values. So we've got our enzyme incubation started right now. Usually it takes something on the order of a half an hour or maybe a couple of hours to get good measurements of enzyme activity. We'll want to take maybe 4 to 6 measurements of the fluorescence over the course of, like I said, maybe a half an hour to 2 hours in order to get good measurements of the rate of fluorescence production. So while we're waiting for that to happen, we should calibrate our standards. So the way we're going to do that is that we're going to take our live sample and treat it just exactly in the same way that we treated our samples for enzyme activity measurements. We're going to add 1 milliliter of the sample and then 100 microliters of the buffer. And then instead of adding the enzyme substrate that produces fluorescence when we're adding when it's in the presence of enzymes, we are just going to add a fluorophore. We're going to add the fluorophore that is produced by the enzyme substrates when enzymes are present. And we're going to do this at different concentrations. I'm just going to show you one. We're going to add small amounts. I'll start out with 10 microliters. We take our AMC stock, which is again dissolved in DMSO. Add 10 microliters. Put a cap on it. Shake it up. And measure the fluorescence. 10 microliters, and the fluorescence is 7,036. The specific, again, we'll want to do this with maybe 5 different concentrations so we can get a linear curve. But the specific concentrations are not the most important thing in the world. The important thing is that the fluorescence values that you measure span the approximate range of fluorescence values that you have in your enzyme experiments. So these enzyme experiments started out at fluorescence values of about 200 or so, and they're going to probably go all the way up to maybe 10,000 or 20,000. So we want to pick out concentrations of standard that are going to give us those maybe 5 or so fluorescence values that span the range from on the order of 200 to on the order of 10 or 20,000. And then I'll show you a little bit, once we've got all of those done, I'll show you a little bit about how to analyze those data.